Meiosis versus Mitosis by Victor and Yegos. DNA replication. The parent molecule has two complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, A with T and G with C. The first step in replication is separation of the two DNA strands. Each parental strand now serves as a template that determines the order of nucleotides along with a new complementary strand. The nucleotides are connected to form the sugar phosphate backbones of the new strands. Each daughter molecule consists of one parental strand and one new strand. Mitosis, early and late prophase. The replicate chromosomes coil up into X-shaped chromosomes, and the nucleolus will disappear. Now the nuclear membrane will begin to break down. Spindle fibers begin to form and stretch across the cell from centrioles that have been moved to opposite ends of the cell. Now we enter late prophase, where the spindle fibers complete forming and the chromosomes attach to those spindle fibers at their centromeres. Metaphase. The tugging action of the spindle fibers pulls the X-shaped chromosomes into a single line across the middle of the cell. Anaphase. The spindle fibers begin to contract and shorten. This action pulls the centromere apart, allowing the sister chromatids to move to opposite poles of the cell. Once they separate, each sister chromatid is considered to be a chromosome. Telophase. One complete set of chromosomes is now at each pole of the cell. The spindle fibers begin to disappear and a nuclear membrane forms around each set of chromosomes. A nucleolus appears within each nucleus. Now there are two nuclei in one cell and the cell is ready to divide. Cytokinesis. Once mitosis is complete, the cell membrane pinches together and the cytoplasm divides. The purpose of mitosis is to increase the size of an organism, to replicate worn-out cells or repair damaged tissue, and to reproduce identical organisms or clones. Mitosis takes place in all parts of your body. Now we move on to meiosis 1, prophase 1. Homologous chromosomes pair up and non-sister chromatids exchange genetic material in a process called crossing over. Metaphase 1. Homologous chromosomes pair up at the equator. Anaphase 1. Homologous chromosomes separate and are pulled to opposite poles by the spindle fibers. One chromosome from each homologous pair is at each pole of the cell. Interkinesis is the stage between cell divisions. During this time, the cell will grow and make proteins as an interphase of mitosis. There is no replication of DNA during this stage. Now we are at meiosis 2, prophase 2. There is one chromosome of the homologous pair in each cell. Metaphase 2. The X-shaped chromosomes form a single line across the middle of the cell. Anaphase 2. Sister chromatids move to opposite poles of the cell. Once they separate, each sister chromatid is considered to be a chromosome. Telophase 2. Spindle fibers begin to disappear and a nuclear membrane forms around each set of chromosomes. Cytokinesis. In cytokinesis, the two daughter cells are separated. The purpose of meiosis is to reduce normal diploid cells to haploid cells, called gametes. One copy of chromosome per each cell. These special haploid cells resulting from meiosis are eggs or sperm.